I'm Mickey Myhre. It's very nice to be here, and thanks to the ACMS and to Alice Blake and the officers and Denise and people who invited me to present this talk tonight. And thank you all for being here. And I would be remiss if I weren't to just offer tip of the hat and acknowledgement for the work that all of y'all have been doing for the last couple of years. I know how hard it can be to practice medicine in ordinary days. These are not ordinary days. I think you know that. And at any rate, kudos to you and thanks for what you do. I hope you find this talk informative and interesting. The bunch I want to try and cover in as brief a time as I can. So if you don't mind holding questions till the end, I'll be happy to answer them, and I can probably carry on all night and all day if you're so inclined. I also wanted to acknowledge the lady who took this photo. Susan Stacy is her name. She's a member of the local Mycology Association, and she took this picture in late September on Harrison Boulevard. We'll talk about the importance of this mushroom in a little bit, but it turns out to be rather a stunning find and an interesting topic of its own. So with that, let's go ahead and get started, all right? When I was a kid in the 1960s, a little older than this boy in the illustration, my sister introduced something to our family from her home economics course that she called PDC. It's called Pleasant Dinner Time Conversation. Which at the time did not include discussion of rock and roll, certainly not rotten mold and fungi. Now, you may have some concerns about asking a pathologist to talk to you after dinner, and I get that, but I promise you that this talk will not disturb your digestion, but may stimulate your interest and imagination, perhaps open some vistas and some intellectual labyrinths that you find as interesting as I very much, to my own surprise, have found it myself. Because tonight, on our stroll through the fungal forest, we're going to be talking about fungi. Fungi are mysterious. They're unseen. They're largely unknown, yet they're critical to life on Earth. They take bare rock and they make soil. They nourish that soil so that plants can grow. They produce spores in such abundance that they influence the weather, help rain to fall to nourish the plants that nourish us. And then at the end of it all, they recycle everything, and I mean everything. So life can begin anew. And they've been doing this for over 1 billion years, maybe twice that long. We begin tonight with the plant. This is a plant, John Muir's glowing pillar of fire plant. It's entirely dependent on fungi for its existence, but you can't see the fungi on which they depend. As Merlin Sheldrake says in Entangled Life, fungi are everywhere, but they are easy to miss. Is there a way we can turn these lights down just a little bit up front? It'd be easier to see the slides if we could. But so as not to miss out too much on the topic tonight, I propose the following structure. 1, just what are fungi? 2, why won't I shut up about them? And 3, how did a rational, reasonable fungaphobe pathologist become a fungophile fanatic? Well, we'll get there. So what are fungi? Well, they're not, as I was taught in college, plants that have lost the ability to photosynthesize. They're their own kingdom or domain, if you prefer, like plants and animals. Animals, everybody here knows, are composed of cells whose cells are limited by membranes composed of cholesterol. They derive their energy by ingesting their food. Plants have cell walls made out of cellulose, and they photosynthesize, create their own energy via photosynthesis. Fungi have cell walls made of chitin, which is the same material from which shrimp shells, crab shells, and so forth are made. And unlike plants or animals, fungi digest their food outside the cell. They secrete enzymes, acids, etc. outside their bodies and absorb the nutrients. What this means is that fungi are never ever separated from their environment. They're immersed in it, and as so eloquently put by Juliana Furci, a Chilean mycologist, fungi cannot nurture themselves without the medium in which they are immersed because they digest the environment in which they live. I want to remind you that fungi are absolutely everywhere on the planet in every ecosystem. Merlin Sheldrake further makes the point elegantly by saying mycelium is the ecological connective tissue, the living seam by which life is stitched into relation. That guy can write, I'm telling you. That may be interesting, but kind of, so what? So why won't I shut up about fungi? Well, I'll give you 3 reasons. One, they're a very large kingdom. This comes as a complete surprise to me, but it turns out that they are probably the largest kingdom of life on the planet. A survey of North American plants and fungi showed that there were about 37,800 fungal species identified and about 18,400 plant species in North America. That's less by far than the estimated 6 to 10 times as many species of fungi as plants as are estimated to exist on the planet, most of which obviously are not known. Now, they may be a large kingdom because they're ancient. It turns out that life began on this planet about 2.5 to 3.5 billion years ago. And if life began 2.5, 3.5 billion years ago, and life on the planet was represented by a 24-hour clock, fungi have been here for about 16 hours of that 24 hours. The Homo sapiens, us folks, have been here for less than a minute on that scale. It gives you some sort of a time scale, all right? Turns out that eukaryotes emerged from their actinobacterial ancestors about 2 billion years ago, and filamentous fungi emerged about 1.5 to 2 billion years ago to take their place on the stage of life, probably as the first true eukaryotes. All of you know, or most of you know, that eukaryotes are organisms that have enclosed true nuclei. You'll hear that term repeatedly. Now, fungi lacking bones, shells, or any hard structures to speak of, and existing as single-cell structures that are about 1/25 the diameter of a human hair, do not fossilize well. But nonetheless, we have fossil evidence from Victoria Island shale that dates to roughly about 1 billion years ago. They've been here a long time. They're probably the first multicellular organisms to venture onto land, and mycorrhizal fungi enabled plants to invade the land. From the very beginning, fungi have worked with other organisms, and we'll see that throughout this talk. Now, plants arose from that line about 1 billion years ago. Interestingly, the cell line from which plants and animals arose appears to have arisen from an ancient organism that's now classified as a chytrid, which is a type of fungus, has a posterior flagella, and as evidence of our lineage continuing from that line, the presence of posterior flagellae on mammalian sperm or animal sperm is said to be one of the holdovers. One of the great ironies to me is that the place I know that you can find chytrids most abundantly on the planet right now are in cow stomachs. There are about 1 billion of them in the rumen of every cow. And these fungal organisms that gave rise to plants and animals now live in the gut of an animal, helping them digest plants. So go figure. At any rate, it was a surprise to me to find that plants were only able to grow on land about 400 or 500 million years ago because of mycorrhizal fungi. Those are the fungi that interface with the roots of plants. Fungi served as root systems for plants for millions of years before plants were able to develop root systems of their own. Now, plants arose on the terrestrial landscape at a time when fungi predominated. This is an artist's rendition of a fungus that's called Prototaxis, I think is how you would say that. But anyway, it's a columnar organism of which fossils have been found that is 9 or 10 meters high, and they were the largest terrestrial organism to ever live on the planet up to that point, and their dominance on the planet lasted for about 40 million years, or about 20 times longer than Homo sapiens has been on the planet now. Now, after plants made out of water, a joint fungal animal line that's called Ocreata continued for about 200 million years, diverging into animals and fungi at about 800 million years ago. Now, again, surprising to me, and this word comes up over and over again because fungi have just surprised me out the wazoo. It's a phenomenally fascinating area, but at any rate, molecular evolution studies show that fungi are the closest relatives to animals. So they're a very large, very ancient kingdom. They're closely related to animals. That's one reason I won't shut up about fungi, but second reason is that they're critically important to life on Earth. They are important to life on Earth now, and they have been for about 1 billion years. Their association with other organisms in particular, especially plants, means that they are and have been critical to ecosystem operations from the very start. Next slide. There we go. The critical role that fungi play in ecosystems is evidenced by the fact that forest soil, which feeds the forest, you've got to realize that, it's about 90% of the living biomass of the soil in forests is fungal. All right? We'll touch on some of this here in a minute when we're talking about soil fungi, but they're in fact crucially important to the maintenance of life on Earth, period. Let's touch on reason number 3 why I won't shut up about fungi, and that's that they are just unimaginably weird. And we will go into that here in a little bit, but for now I'd like to begin retracing my steps with where I first began to learn about fungi, which was in college. I graduated from University of Texas. It's not their fault. The first biology class I took was at University of Houston, and again I learned that fungi are plants. Lost their ability to photosynthesize. Well, that sounds like a stupid idea, but it's an idea that comes from a guy by the name of Ernst Haeckel, who in 1866 proposed 3 domains: plants, animals, and protists. Protists are microorganisms. He classified fungi as plants, and they remained classified as plants until the late 1960s, when Robert Harding suggested a supergroup, this Ocreata, containing both animals and fungi. It wasn't until 1990 when a scientist by the name of Carl Woese discovered that relations between organisms could be elegantly deciphered by means of studying ribosomal RNA genes, a process that led to what's called DNA barcoding, a method that now seems to be the method of choice for identifying every kind of species you can think of, from fish to fungi and everything in between. So what are fungi? What do they do? Well, they're microscopically small organisms, as I mentioned. They digest everything. As lichens, they digest rock to make soil. They digest wood turns out that fungi are the only organisms on the planet that can digest cellulose and lignin. They didn't develop this ability until 360 million years ago. Up until that time, all the plant material on the planet just piled up miles deep. Net effect of which is that that's what developed turned into coal, oil, gas, all our petrochemicals. But the fungal ability to digest cellulose and lignin ended that deposition, ended the Carboniferous age, and explains why we have only limited amounts of these fuels. In addition to normal things, fungi can digest, well, I said everything. Plastic. There are some Yale researchers who, I forget where it was, I think it was Ecuador, but somewhere in Central America, they identified a couple of species of fungi that can digest at least certain types of plastic. So clearly, fungi are metabolically diverse and they grow everywhere. So how do you classify them? You find them on the floors on sea floors. You find them on the frozen valleys of Antarctic, in deserts, and on every possible niche of this planet, there are fungi in or on them. So how do you organize, or how do you classify these things? Well, right now they're classified in three main phyla: the Glomerulomycota, which are the soil fungi, possibly the most important group and the least well understood. They're obligate symbionts, which means that they require their hosts in order to live. You cannot culture them independent of their hosts, for which reason only about 200 species have ever been identified. But they've been around for about 500 million years, and Glomerulomycotan fossils have been found in mid-Ordovician rocks in Wisconsin 460 million years ago, before the first vascular plants ever arose. Why are they important? The reason that they're important is they make a molecule called glomalin, which is the most important molecule you've never heard of. At least that's my read of it. Glomalin was only identified at all in 1996 by a USDA scientist by the name of Sarah Wright. Its protein structure has still not yet been worked out, though the soil consists of about 30% glomalin. It's a dark brown molecule, and I've seen studies where they separated the glomalin from dirt, and guess what? Dirt without glomalin is kind of a light tan. The thing that makes dirt brown, glomalin. Without it, plants don't live. Also, it turns out that glomalin may account for a third of the Earth's soil carbon, and it contains more carbon than all the plants and the atmosphere combined. That's big. As a glycoprotein, meaning it has sugars on the surface of the protein, it's a sticky protein. It provides a sticky glue that's important for soil structure, allows for water retention, erosion control, and where did that go? Okay, erosion control allows for the passage of air, roots, and water through the soil. It basically makes soil arable. You can grow plants on it because glomalin's there. Second major phylum is the Ascomycota phylum. These are your molds and yeast. There are about 64,000 of these species that have been identified. This includes the Penicillium mold, the morel mushrooms, Canada, and then everybody's favorite probably is Saccharomyces cerevisiae, which gives us bread, wine, beer. Unsurprisingly, it was Saccharomyces cerevisiae, which was the first fungal organism that had its entire genome sequenced in 1996. And molecular evolution studies show that Saccharomyces cerevisiae originated in Eastern Asia about 15,000 years ago. Further studies of yeast DNA have shown that about 20 to 30% of yeast genes have human homologs. Study of these yeast genes have allowed the elucidation of a number of human diseases, particularly Friedreich's ataxia, ALS, mitochondrial, and other metabolic diseases. But they've turned out to be a hugely important research tool. The final phylum we'll discuss is the group, the Basidiomycotes. There are about 32,000 of these that have been identified. These are the mushroom fungi, basically. These are the ones that produce mushrooms, and generally if you mention fungi to people, they think of fungi, but it turns out that of the 1.5 to 5.8 million species of fungi on the planet that are projected to exist, only about 10,000 species produce mushrooms. Most disperse their spores by means of means other than mushrooms. Of the 10,000 mushrooms that are the 10,000 fungi that produce mushrooms, only about 400 are poisonous. 20 of those are commonly found. 6 are lethal, and we have 3 of these in Idaho. The first deadly mushroom we have in Idaho goes by the name of deadly Gallerina. This is a picture. I don't know if I can walk all that way with this tether on me, but that kind of orangey looking guy right there, that's a Gallerina, that's deadly Gallerina. The ones around it, those are psilocybe mushrooms, the magic mushrooms. Be careful. Know your mushrooms and examine them carefully. And the problem is because these mushrooms are illegal and they're often harvested at night, some people get in trouble with that. It takes about 4 of these deadly Gallerina mushrooms to prove lethal. So bear that in mind. The second deadly mushroom we'll talk about is the Amanita ocreata, the destroying angel. It's a pretty picture here of one. I would point out that if you want to identify a mushroom, dig up the base, the whole base, because you see this little sheath around the bottom of the mushroom? That's a hugely important feature for identifying all of these amanitas that are really deadly. So at any rate, you dig these up and maybe somebody can tell you what they are. Eating a single mushroom of this species is enough to kill you. My favorite, the death cap mushroom. Now, where did my tech go? Come on, wake up, everybody. Death cap. Death cap, and the screen dies. Amanita phalloides. This is that picture I showed you before. Again, this is taken by Susan Stacy, a mycology member of the local group on Harrison Boulevard. And get this, not long before Halloween. All right? It's a big, beautiful mushroom, and it tastes good. One of the reasons I'm really excited about it is that it had never been identified in Idaho prior to September of this year. It had never been identified this far inland before September of this year. This is a mushroom that's by far the deadliest of any of the deadly mushrooms. It's responsible for 90% of the mushroom poisoning deaths worldwide. And one mushroom contains enough of this amatoxin, the deadliest poison in it, to kill 4 people. Now, I took this photo the night that Susan brought this mushroom in, and salient features to note are this little, they call it a vulva, this little membranous structure at the base there. The white gills and this kind of golden, greenish, metallic kind of sheen. Those are all salient features, but at any rate, don't eat any mushroom that you don't know really, really well. And again, the problem with this mushroom is it tastes good. That feature was probably used by Emperor Claudius's wife to poison Emperor Claudius. It tastes good. You don't get sick for about 3 days. Then you get, like a GI thing, vomiting, diarrhea. You get better, and you think, it's just a little bug. And a day later, you look in the mirror and you go, why am I yellow? You go see the doc and your liver's dead. And you either get a transplant or you're dead. Boom. Now, I never autopsied any patients who died of mushroom poisoning. I autopsied an absolute ton of people who died of opportunistic fungal infections, particularly at MD Anderson, and afterwards in chemotherapy patients and so forth. You guys all know this story. This is a picture of, I don't know this for sure, but it looks like Aspergillus niger in lung tissue. Aspergillus niger is just ubiquitous in the environment. Everyone, it's absolutely everywhere. You can't get rid of it. In patients post-chemotherapy, any kind of bone marrow transplantation, any kind of immunosuppression, they're set up for this stuff. But I was definitely a pathologist fungaphobe. I'm proud of it, looks like. My professional impression was that because humans are so closely related to fungi, they being eukaryotic as opposed to prokaryotes like the bacteria that we treat with antibiotics and stuff, that they're a lot more closely related to us. And so what it turns out is that they're a lot tougher to kill without killing the patient. The things that make fungi sick in terms of therapy do harsh things to people. I would call you, you guys all know this story. You all have been exposed to amphotericin, and I don't know what the current antifungals are like, but they used to call it the Red Death or amphoterrible on the ward. It's tough on people. But conversely, because fungi are closely related to us, they live in just teeming masses of bacteria, viruses, and other stuff that's trying to eat or kill them. The things that fungi use to protect themselves from pathogens by which they're surrounded can help humans defeat the diseases that are caused by those same organisms. So what's good for fungi can be good for humans. This is an interesting photo to me. This is, case in point, I guess, is penicillin. We've got Sir Alexander Fleming here on the right with the first patient who was successfully treated with penicillin in 1942, I think it was. Fleming actually identified penicillin in 1928, and they were unable to develop a strain that produced penicillin in sufficient amounts to provide for commercial production. I'd also back up and say that the only reason that it was discovered is that Fleming was slipshod in his lab work. He was notoriously sloppy, and he walked off and he left a plate of Staphylococcus growing on a petri dish and forgot to put the top back on it. Came back after the weekend and there was fungus growing in it, and it was killing the staph. He isolated the fungus, put it on bacteria, and found out the bacteria died, and he called it fungal juice. Now, I'm glad he changed the name to penicillin, I guess, but at any rate, the first patient who was treated with penicillin was actually an English bobby who was pruning his roses, and one of the branches popped up, hit him in the face, caused a staph infection in his skin that spread and developed cellulitis, and they wound up enucleating the guy's eye. They removed his eyeball surgically because it looked like he was going to die from this infection. And when that didn't work, they thought, well, we have this fungal juice. Let's see if it'll maybe help this Bobby because he's going to die. They squirted it in him, and he made a remarkable recovery, and they didn't know they'd have to give him more than one dose. They only had one dose, and he died. Well, in 1942, they had enlisted the help of American mycologists in hope of interesting the American pharmaceutical industry to come up with a means of producing penicillin, which they had not been able to do up to that point. And one of the lab techs Mary Hunt, found some golden mold growing on a cantaloupe that she was preparing for her family. She cut up the fruit, served her family the fruit, and took the moldy bit to work and said, let's try this. And it turns out that that strain of penicillin from the moldy cantaloupe was a species they termed Penicillium notatum. She found this moldy cantaloupe in a market in Peoria, Illinois, for which reason here just, I think last month, Illinois named Penicillium notatum the state fungus of Illinois. Now, Alexander Fleming got a Nobel Prize in 1945 for his work on this. Mary Hunt, who brought the fungus to the world, got the unfortunate, the unfortunate nickname of Moldy Mary, and was promptly pretty much forgotten. Another important medicine we get from fungi is Taxol. Isolated in 1971 from Taxomyces andreanae, a fungus living in yew trees. If you look at anything about Taxol, they say it comes from yew trees. It doesn't come from the trees, it comes from the fungus that lives in yew trees. So there. It's an effective chemotherapy agent, it's $1 billion a year drug. Cyclosporin's another one. That was isolated from Topocladium inflatum by Sandoz scientists from soil in Norway and Wisconsin. Sandoz evidently sends scientists around the world digging up dirt and looking for fungus, and you know why? Well, it works. Turns out you guys know a lot more about the use of cyclosporin than I do, but it opened the door to liver transplantation and probably all other organ transplants. Final example are statin drugs. Those are isolated from Penicillium citrinum. The statins are the second most commonly prescribed drug in the world. At least $25 billion a year. Who knows? I know lots and lots and lots of statins sold. But one thing I'd point out to you is that oyster mushrooms, which you can buy at your local markets, either like Roots, the zero waste market on Chinden, or the Co-op, both have oyster mushrooms all the time from a local mushroom farm in Parma, Groves country. Nice guy, Mason Grove, run it. But oyster mushrooms contain up to 2.8% lovastatin, the same chemical, absolutely the identical chemical is present in oyster mushrooms as in the prescription medicine. Okay, so fungi are ancient. They're a huge kingdom. They're important. They provide a lot of medicines. And how did I go from this to this? How did I become a fungophile fanatic? I mean, how in the hell did that happen? That, by the way, is an oyster mushroom. Whoops, pointing at me. Oyster mushroom on my head. Clearly, something changed. What happened is that I attended a mycology workshop, a weekend workshop hosted by a fellow by the name of Peter McCoy. He wrote this book, and he holds these radical mycology workshops around the country. He drives a little beat-up pickup truck. He's outfitted so he can sleep in the bed, does these 3-day workshops, and drives to the next place all over the country. This is a mission for this guy. And he does a really good job. A couple of things in that course just absolutely blew my mind. One was the fact that maybe 5% of the fungi in the world have yet been identified. At least 95% are unknown. I mean, imagine telling a young kid who's like a National Geographic Explorer Club that 95% of the Earth is yet to be seen. I mean, it was that kind of excitement that I felt when I heard this, and I thought, my God, I've studied biology my whole life, and who knew? Well, at any rate, he showed a video of the growing tip of a fungus, the mycelial tip. There's the part of the mycelium that is essentially, like I tell lay or non-scientific audiences, it's like the Amazon packing and shipping center for products that are needed to grow the tip of the mycelium and be able to extend. Well, at any rate, this video was fascinating. And I mean, it was going, a lot of things going on really fast. And then he started going through the things that aren't there. But basically, none of the organelles I'm used to in all of cell biology are there at all. Golgi apparatus, the organelles, the structures, we don't have a clue how this works. And this is the largest kingdom on the planet. We don't know how they even grow. Well, that got me a little excited, got me a little curious. But I figured, I left that workshop and I thought, well, man, if we know that little about them, they can't be very important. I mean, seriously. How can science have left such a huge, important area undiscovered if they are important? I can't figure they couldn't be very important. But when I looked into it just a little bit, it turns out that there are, like I said, 6 to 10 times as many species, fungal species, as plants. That fungi comprise about 25% of the entire biomass of the planet. The largest organism on the planet here is seen in an aerial view. It's located in Myhre National Forest in eastern Oregon. It's a fungus, and Armillaria is the genus. It's also known as honey mushroom. The honey mushroom is a tree killer and a meadow maker. It kills the trees, allows the grasses and the plants, the short plants that are otherwise choked out by the trees, to come in and grow, and it begins a whole new cycle of life in the wild. This single organism is at least 2,200 acres in size. It's been estimated to be as old as 8,650 years old. And they've tested the DNA from one end of this thing to the other, from the middle to the edge. It's identical. It's one organism is that big. Well, still, knowing that fungi are as large as that, I still didn't expect that they would produce the amount of spores that they do. Fungi produce about 50 megatons of spores a year, which is equivalent to about 500,000 blue whales. Now, if we had 500,000 blue whales floating around in the atmosphere, we'd know about them. We'd notice them, but we don't see a thing because they're too small. If you flattened all those spores out and laid them side to side, put them flat on a piece of land, they would cover the entire continent of Africa every year. That's how many spores there are in the world. Jennifer Doudna, who I think she got a Nobel Prize for her work on CRISPR technology. If she didn't, then she's gonna. But anyway, according to her in her book about that project, she makes a statement that fungal spores are the second largest biomass on the planet, second only after bacteriophages, which surprised me. At any rate, fungi produce enough spores that they influence weather. The way they do that is the spores are there, again, in such profusion. They allow water droplets to, they provide a surface for water droplets to condense on that allows droplets to form, clouds to form, and then rain, snow, and so forth to fall. Their importance to the ecosystem, then, has always been important. But at any rate, they produce so many spores that they influence the weather, and they've been important to the ecosystem forever and ever. Now, the vast majority of plants on the planet rely on mycorrhizal fungi. Mycorrhizal, just myco is fungus, rhiza is root. It's the fungus that live in and around root systems of plants. This works out well for both parties because the plants provide glucose and lipids to the fungi. The fungi provide phosphates, minerals, and water to the plants. They have a really effective and interesting barter system they set up that I think would be really interesting to study as an economic model, essentially. But at any rate, when you walk in the forest, all of whose plants rely on those mycorrhizal fungi, under every footstep, you've got about 30 miles of mycorrhizal fungi. The total length of mycelium on the planet has been estimated to be a mind-boggling number. I don't have any. It's about the diameter of the Milky Way galaxy, right? You're talking hundreds of trillions of miles. And they're all connected. Not just to each other, but this is a cross-section of a root section. Little green bits, these are mycorrhizal fungi in a root tip, and you can see how dense they are. The green bits are the fungi, and the clear bits are the plant cells. And there can literally be hundreds of species of mycorrhizal fungi living in a single plant. These fungi are also termed arbuscular mycorrhizal fungi or AM fungi. Arbuscule means little tree or little bush or something. You can see where that would kind of come from. If you use your imagination, that could be like the trunk of a tree, and these are branches, and these are roots. But at any rate, you get the picture. They're in the roots. The blue-green part's the plant, the red part's the fungus. Now, I don't know if I'm really sure of this, but I have read really highly regarded experts make this statement unconditionally that no plant grown under natural conditions has ever been found without these fungi. Certainly, it's the predominant fashion by which plants prosper on this planet. These mycorrhizal networks allow trees, for instance, to communicate with each other through the mycelial network. What they'll do is they will send out signals via the mycelium that prevent their seeds from sprouting when the mother tree is under attack by pest or disease. And when this disease is passed or the pest has moved on, they allow their seedlings to prosper. They also send out nutrients to their seedlings when they're too small to reach the light themselves. And it's interesting, it's not just their own species they do this with. There's a lot we've got yet to learn about these things. But they clearly provide connections between plants, a structure and a system that's been termed the Wood Wide Web. Popularized, I think, by Science Magazine's publication of Suzanne Simard's groundbreaking work in the area. We'll see a picture of a slide of her work here in this next thing. This is a really confusing slide, but I'm not going to even begin to try and dissect it in any detail, but all the little green bits, these are trees. They're all different kinds of species. The lines between them are the mycorrhizal, are fungi of a species of mycorrhizal fungus connecting the trees. And as you can see, the trees are connected to each other. They're connected by multiple different mycorrhizal fungi, and it's complicated as hell. Now, this is a very simple sort of study that she did. She had to study a defined range of mycorrhizal fungi, because if there are hundreds in every plant, there's no way you could end your study. But at any rate, she used radio-labeled carbon 13 and 14 to see where that carbon went when she administered to one tree, and she could trace it across the path and to great distances. At any rate, she demonstrated that each of the trees was connected via mycelium, often by many different varieties of fungi, to many different plants, and in a huge network. As part of that work, Susan Simard demonstrated that the trees take CO2 from the air and store about 70% of it in the soil, again in our friend glomalin. This points out the critical role that fungi play in sequestering carbon, and particularly in its potential in regulating climate. Because that role is so important, the role of fungi in addressing climate change, in my view, I don't think can be overstated. It's not just addressing climate change that makes fungi important. You see here is a picture of what your store shelves look like in the absence of fungi. There's going to be no bread, wine, beer, fruit, vegetables, and certainly no animal proteins. And without fungi, essentially all life other than bacterial ends. Unfortunately, our current agricultural practices aren't real fungal friendly. The application of fungicides and pesticides and all those kinds of good things, inorganic fertilizers, etc., harm the interaction between fungi and plants, and it interferes with their ability to form those relationships in a meaningful way. Further, the use of antifungal chemicals has led to some absolutely devastating diseases. Right now, the deadliest disease ever known on the planet has led over 90 species now of amphibians to extinction and threatens God knows how many more. It's interesting, it's a chytrid type fungus that's doing this, but at any rate, rice blast fungus resistant to antifungals spoils enough rice to feed 60 million people a year. And the one that hits me emotionally, because I like bananas, is there's a fusarium fungus that drove the species of banana previously planted all over the world to extinction, is now infecting the Cavendish strain that had shown resistance. So enjoy your bananas while you can. You know what else happens when ag plays poorly with fungi? Remember how the soil bacteria, the Glomerulomycota fungi, store carbon in glomalin? Well, if you kill the fungi, guess what happens to the carbon? It goes up. So, turns out that our current agricultural practices, according to soil scientists in Corvallis, Oregon, releases about 25 to 35% of the soil carbon. And again, per these same authors, more than, quote, all types of transportation combined. That's taking every internal combustion engine in the world, every coal power plant, every damn thing you can think of. None of it comes close to the CO2 that's released just by our agricultural practices. Good news is that it's reversible, and with minimal tillage and less of these noxious chemicals thrown on the ground, rates of carbon deposition can increase dramatically. Other industries outside of agriculture have adopted the use of fungi in a very, very big way. 60% of enzymes used in industry come from fungi. Those stonewashed jeans some of you might be wearing, that's a fungal enzyme. Your whiter than white enzymes in your washing powders, fungal enzymes. 15% of vaccines are developed in largely yeast. 80% of the citric acid in, I don't see anybody with a bubbly drink, you know, like soda, but 80% of the citric acid used to create the carbonation in fizzy drinks comes from, guess what? Aspergillus niger, our good friend that was in the lungs and you saw back there. In addition, increasingly, mushrooms and fungi are being used therapeutically and tested in medical trials. I term this section medically interesting mushrooms because I don't want to come off as like I don't know, this is mainstream medicine, although it's really close to becoming that in a lot of areas. We're going to start off looking at a study, and we'll start at 30,000 foot level, looking at a study out of Singapore that I just think is fascinating. This is a 6-year study of 663 patients who are older than 60 years old. They studied them for a 16-week period and took note of what they ate, and particularly if they ate mushrooms or not. They didn't control for the type of mushroom. This is Singapore, and Asians eat a whole lot of different kinds of mushroom. They didn't even try. Just mushrooms or not mushrooms. But it turns out the patients who ate mushrooms more than 2 times a week had roughly half the incidence of mild cognitive impairment versus the control group. I'll say that again. Ate mushrooms three or more times a week. Those people had half the incidence of mild cognitive decline or impairment. It's basically early dementia. They postulate the effects are due to ergothioneine, an amino acid that's been proposed as a new vitamin, since it's required by the body and it's not produced by it and it has to come from an exogenous source. The fungi mushrooms are the highest, or it's found in the highest density or highest concentration in mushrooms. At any rate, whoops. And I want to point one thing out here. You're not going to be able to read this from where you are, but this association with the mushrooms, you think, well, okay, this isn't maybe it's not well controlled for, let's try some of the things they looked at. Age, gender, education, cigarette smoking, alcohol consumption, hypertension, diabetes, heart disease, stroke, physical activity, and social activities. Seems like a pretty good study. Me, I like to eat mushrooms more than a couple of times a week if I can. If I can remember. Yeah. One of the more interesting and well-studied mushrooms whose neuroprotective effects have been studied pretty well is lion's mane mushroom, the Latin name for which is Hericium erinaceus. The lion's mane mushroom has long been known to cause nerve growth factor release in petri dishes, in vitro. It's been shown now very convincingly to cause nerve growth factor release in vivo. Now, in rodent studies, I find this really fascinating. There are some great, I don't know if they're great or not, but they have rodent models for Alzheimer's disease. They're bred to develop it, and they do terrible things to them, and they make them develop dementia. But the rodents that have this experimental Alzheimer's, when fed lion's mane mushroom, it causes a diminution in the size of the plaques. The cognitive scores of the test animals improve while they are taking the lion's mane in their diet, and their cognitive scores decrease after cessation of feeding the lion's mane. Maybe it's helping. Who knows? Human studies out of Japan are even more interesting to me. Next slide, there you go. In this study, they studied a far smaller group, 30 patients with mild cognitive impairment between the ages of 50 and 80 years old. They followed them for 20 weeks. And the cognitive scores of these patients improved during the 16 weeks that they were taking lion's mane. They gave them 250 milligrams of lion's mane powder capsules or tablets 3 times a day for 16 weeks. Stop that. For 16 weeks. They then stopped giving them the mushrooms, and they tested them again. 4 weeks after the termination of taking the lion's mane mushroom, their cognitive scores decreased to a statistically significant level. Again, seems like it might be helping. Flip side of that coin is that, to my knowledge, there have not been any serious side effects associated with lion's mane administration. So, guess what? I've grown lion's mane. I take a lot of lion's mane, and it's a delicious mushroom. It's also quite often available at the Roots Zero Waste Market on Chinden and in the co-op. But at Roots, they have it all the time, and it's produced locally. Nice guy too. Compounds called Erinacines, of which there are at least 15 different varieties, are thought to be responsible for this. Those come from the mycelium, or the body of the fungus. There's another class of compounds called carcinogens, of about at least 11 of which come from the fruiting body of the mushroom that looks a lot like a lion's mane. It's also called a puffball mushroom. They're thought to be responsible for the neuroprotective action of lion's mane by a variety of mechanisms shown here. We're not going to go through it, but all these are good things. The things that are going up, you want up. Nerve growth factor, you want up. Microglial activity, which are the kind of stuff, the cells in the brain that make for scars and nastiness and plaques and stuff, that goes down. All the things that you want to have happen to improve Alzheimer's, seems like lion's mane does an awful lot of it. Well, experimental studies thus far, and I have references. I didn't put together an extensive reference list. I can certainly get references to anybody who wants them. But at any rate, experimental studies have shown, or at least suggested, protective effects in ischemic stroke. It's been shown to be helpful in depression, said to help improve, possibly prevent Parkinson's disease. It's been used in therapeutic trials for neuropathic pain, and again in depression. And for a mushroom that, again, to my knowledge, has not had any serious side effects, these are significant potential upsides. Another mushroom that I find really interesting, and one we keep turkey tail tea in the fridge at home all the time. So it tastes good. It tastes a little bit like sarsaparilla or root beer or something. But at any rate, it's a beautiful mushroom. Grows on hardwood, so you don't find it that much around here. But it's Trametes versicolor, also called Coriolis versicolor. Now, turkey tail mushrooms are widely used in Japan and China as a cancer chemotherapy adjuvant. It's a several $100 million a year market there. The interest in this mushroom has only surfaced really relatively recently. The NIH sponsored a $2.1 million study. I don't remember recently, by Paul Stamets. It showed significant anti-neoplastic potential as an adjuvant agent. It also showed significant antiviral and anti-inflammatory effects, for which reason researchers in Italy and St. Louis University of Medicine decided to study turkey tail mushrooms in combination with lion's mane mushroom on traumatic injury-induced Parkinson's disease. This study, published in 2021, found that, quote, Parkinson's disease-related molecular events may be triggered on traumatic brain injury, and that nutritional fungi such as Mycelium erinaceus, lion's mane, and turkey tail may be important in the redox stress response mechanisms and neuroprotection, preventing the progression and neurodegenerative diseases such as Parkinson's disease. Again, I've got that reference. This is a picture I'm a guy who spent years looking through a microscope, so I can't help myself. I had to have one picture of some slides, but basically they took these poor mice, and there are evidently some standardized regimens by which you whack mice on the head till they develop Parkinson's disease. and when you make slides of them, guess what? The slides are pretty much blank, as is their brain. It's like, well, when in these slides, the more dots, the better. All right, the dots are nuclei of cells that you want in the brain, and this is turkey tail mushroom by itself. This is lion's mane plus turkey tail, and this is better than that. That's the punchline of that story. At any rate, it may be important in preventing the progression of Parkinson's disease. This is an area I'm paying very active interest. I have a very active interest in following, and like I said, we eat lion's mane mushroom and we eat capsules of lion's mane, turkey tail, good stuff. Why not? Another mushroom that you couldn't have missed in the last couple of years is the psilocybin mushrooms, the magic mushrooms. Been everywhere. The Newsweek magazine, you may have seen on the stands. There was an article in the medical literature, the headline of which read, psilocybin therapy 4 times as effective as the conventional antidepressant therapy. I mean, it's been everywhere. So the numbers of studies going on utilizing psilocybin right now are just enormous. As of this summer, there were 67 clinical studies sponsored by the National Institutes of Health. I'm going to point out that synthetic psilocybin has been used in all of the clinical trials at Hopkins and in the research centers all over the world at this point. But Roland Griffiths of Johns Hopkins evidently is considering further trials using the whole fruiting bodies of mushrooms, and is talking with a guy, not really a friend, but he's an acquaintance of mine, owns a mushroom farm in South Carolina, and he's currently growing 14 varieties of psilocybin mushrooms in Jamaica, possibly for use in clinical trials in Johns Hopkins. Studies thus far have shown highly promising results in depression, PTSD, addiction, OCD, pretty much anywhere where a reset of the default mode network of the brain, which is kind of the brain's treadmill, where it's running dysfunctionally, needs a reset, seems to really work well. Of all the studies out there, there's one that did catch my attention that I'm going to spend just a little bit of time talking about, because it looked at neurogenesis and synaptic connections in this is a rodent model, given a single large dose of psilocybin. What they did is they gave these rodents a comparable dose of chemical psilocybin, comparable to the clinical trials that they're using on a per body weight basis. Right now in clinical trials, I think at Hopkins, they're giving them like 25 milligrams of synthetic psilocybin, which is roughly, since the psilocybin mushrooms contain roughly about 1% psilocybin, that would be maybe 2.5 grams of the dried mushrooms, something like that. But at any rate, they gave one large dose of psilocybin to these lab rats. They found that single dose of psilocybin led to about a 10% increase in neurogenesis. This is new nerve growth in their brains. In addition, the synapses, the connections between the nerves, were 10% more dense than the controls. These effects occurred within 24 hours of the administration of the dose and persisted, and this is in a mouse model, 1 month later, which is a significant period of time for mice, which they don't live very long in a lab setting, I know that. I've done some of those studies. This suggests to me a possible mechanism underlying the observed efficacy of psilocybin in diverse test populations. For those who want to check the reference, I put the DOI thing there. I can get it to you if you want. The URL on the slide works, and you can check this link for yourself. They found that it ameliorated stress-related behavioral deficit, and again, the structural remodeling, the increased neurogenesis, and the denser connections occurred very rapidly and persisted. I would just parenthetically add that I recently read studies that that looked at some human psilocybin trials in depression, and there they found about 75%, 70-something% of patients who came in with clinical depression responded extremely well to a large dose of psilocybin. Some studies use one dose, some use two. Some places, the ones that did two would administer a low dose the first time, wait a few days so that the resistance wore off or weak or whatever, and then they repeat the experience. But at any rate, it's a limited exposure over a limited time, and they found that there about 70% of the patients that responded had therapeutic benefits for 9 months to a year is the period that I recall. At any rate, it's a promising medication, and I don't think we have any psychiatrists in the audience. If you do, I'd be interested to hear what the view of that field is on this new research, because to me it's really interesting. Well, the psilocybin mushrooms would cause neurogenesis is pretty weird, but now we turn to the reason number 3 that I just can't quit talking about fungi. Yeah. Okay, we're almost at the end. We're almost at the end. They're unbelievably weird. Try Chernobyl. In Chernobyl, where fungi may be as tough as the people, the fungi at Chernobyl were found growing towards radiation and using it as a food source. They can be so specialized, there's a Lobule's fungus that grows only on the left wing of certain beetles. The split gill fungus, a pretty little thing, has more than 25,000 sexes. That's basically as many people as you can fit in the Albertson Stadium at BSU. You think we got gender issues? I mean, I can't even imagine. At any rate, no discussion of weirdness in fungi is complete without cordyceps fungi. This picture is, I would point out to you, upside down. This is the underside of a leaf. What cordyceps does is it infects, in this case, an ant, causes it to climb up to the highest point it can. It causes it to bite down on the stem or the central vein of the leaf, digest the jaw muscles of the ant, and then a pretty little mushroom pops right out of its head or its butt or its thorax, right over the pheromone-laden trails that the ants use to follow their way back to the nest. So they're dropping their spore bombing the ants down below them. It's just brilliant. Massive spore fungus is even weirder. Drug castrated and ready to mate was the article title. It's one thing I read. Fungus makes an amphetamine that's popular in the Middle East and the Horn of Africa called khat, and psilocybin. And no, you can't get high by eating cicada butts, but it makes their butts and their genitals drop off, all right? And incites furious mating behavior on their part, and they try and mate with every cicada they can. Male, female, any kind of cicada, they're all over it, just trying to mate with it. Just spreading spores like crazy. Well, with an organism so poorly understood, so important, and so weird, I had to do something to study it. So I set up my own little DNA laboratory a handful of months ago. This is an example of a DNA barcode. The width of the code corresponds to the number of copies of A, T, G, or C in the genome being studied. Looks like a UPC barcode, that's why they call it DNA barcoding. Well, remember this puppy? The Amanita phalloides mushroom. I took that mushroom to our little baby laboratory and ran it through. And lo and behold, it shows a 99.85% match with Amanita phalloides. We have the first absolutely definitive identification of this mushroom outside of a coastal area, I think in America. The editor of Fungi magazine asked me to write up an article about it and to report it. He's an Amanita expert, and he's super excited about it. It's the farthest inland this deadly mushroom has ever been reported. Now, I hope to identify maybe some new species, and then didn't have any idea this was going to be happening, but wanted to definitively identify the mushrooms and fungi in this region because nobody's done it. Why not? I'll do it. Well, this might be my final professional foray into studying the beauty and complexities of nature. And as Alexander Humboldt said in 1845, each step we make into the intimate nature of knowledge of nature leads us to the entrance of new labyrinths. Melvin Sheldrake provided a really good quote of that, as saying that we are standing at the entrance of one of the oldest of life's labyrinths. E.O. Wilson, who died in December of this last year, a great naturalist biologist, said that his work took him to the, quote, very heart of wonder. And fungi, to me, present at the dawn of life on this planet, never, never, ever fail to incite that sense of wonder in me. My hope is that you can now see that these fungi, on which we are as dependent and as this glowing pillar of fire plant, on the fungi we can't see and don't understand. I hope that this talk has opened some of these labyrinths to you into this very heart of wonder living everywhere around us. I want to thank you for the time that we've spent kind of strolling through this weird forest together. And if you have any questions, I'd be happy to answer them now. Thank you. Yes, Amy?
How much of our microbiome, whether it's PIs,
That's a good question I don't know the answer to. I'm be real honest with you. I know that we got fungi everywhere. The vast majority of our biome is bacterial. The role of fungal organisms in our biome is, I don't know that much about. The biome itself, I think, is a great natural, that's one part of nature that's just begging to be explored by science. I'm going to make a quick pitch. My wonderful wife, Gloria, put together these packets of lion's mane cookies. They're made with lion's mane by the author of our youngest daughter wrote a book called Just Mushrooms. It's a cookbook, mushroom-centric cookbook, and the chef who worked with her cooked these mushrooms, baked them, yeah, whatever. Anyway, pretty little things. Mushrooms are free. If you want a book, talk to Gloria. They're $19, I think, or you can buy them on Amazon. And I'm told I didn't have to disclose that at the beginning of the talk because this isn't for CME. So, hey, come by, everybody. Nobody's making any money on it, but it's a passion project. Yes? Wonderful.
Comment since COVID, we've been seeing a lot of interesting fungal infections in folks who need company. So, for example, invasive glycositis, we normally see in immunocompromised patients. We see immunocompetent folks that operate on the few who have no other evidence of immunocompromise. So that's kind of been an interesting, in addition to knowledge, seeing that those of you recently.
The question is, where the comment is that they've seen opportunistic fungal infections in COVID patients, and the question was raised, well, maybe had they been on previous courses of antibiotics? This is an association I hadn't heard of, and I'm not in clinical medicine and distantly remote from it these days. So that's an interesting, Yeah.
It's interesting, you know, that they're more stable. So, for example, in a patient with pure neural spread of nuclear, but if you we resected the kind of the disease that was resectable, but then there was still a bunch of disease in the camera sinus that wasn't really treatable. She made and just made stable. We all thought that this was going to go down here pretty fast. I didn't know what to do with her, and neurosurgeon didn't. So it's interesting. They're more stable. These are more stable infections. They're clearly based on the.
Well, that's, I think, my secret nightmare scenario is if we develop a really pathogenic fungus that's easily transmitted human to human, we're in deep trouble. Because we don't have any good antivirals either, but my God, fungi, when they get, as you know, into the human body in an invasive form, boy, they can do some awful damage awful fast, and they're real hard a lot of times to get rid of. So it's interesting, and I'm glad that you shared that with us. I'm keeping an eye open for that. Any other comments or questions? Well, thank you again. Very much appreciate it. Thank you. And go get your cookies. The cookies are free. The cookies are free. The books, if you want one, are $19. So there you go.